Great. I think we can get started. Hello and everyone. Welcome to today's OTS webinar. Today we're featuring our speaker, Rebecca Miles, who's actually the president-elect for OTS for 2026-2027. She has a really interesting career trajectory, and she's great to talk to. So if you get to meet her at the OTS in person, do say hi. She'll definitely say hi and be willing to chat. So I'm going to let her introduce herself and tell everyone about her story in science.
Well, thank you so much, both to you, Erin and Ronald, for the invitation to be here today. And good morning, good afternoon, good evening, wherever you are joining the webinar today. So I am the incoming president of OTS, and I hope this is an opportunity for some of you to get to know me, as I feel like I'm a little bit of an unknown entity. These are my disclosures. Currently, I'm the CEO of Raina Consulting. I'm on the scientific advisory board at Alloy Therapeutics, and I retired from Lilly last year as a senior director in genetic medicine. So I'd like to give you a background of my career journey and how it co-evolved with the emergence of genetic medicines and how it brought me to OTS. Since I recently retired, this exercise was a little nostalgic for me and fun to reflect on where the industry was when I started my career. And so I hope you pardon that if there's a bit of a nostalgic tone here and there. I'd like to take the time to highlight the work and the impact of the society, and then finally transition to some thoughts on emerging trends and challenges and the opportunities in the field. And hopefully that sets the stage for discussion and questions at the end. So after my undergraduate degree in Indiana, I moved to the East Coast in beautiful Williamsburg, Virginia, to earn a master's degree in molecular biology from the College of William and Mary. And I finished my PhD at the University of Indiana at the School of Medicine. And from academia, I went straight into drug discovery. Really, from the moment I took human anatomy and physiology in high school, I became insatiably curious about how the body worked and what went wrong to cause disease. And when I realized the joy of doing research and that it was possible to make discoveries that would help sick people get well, I knew it was a perfect calling for me. And the greatest joy in my journey has been my family. So after the last several years, I've had many opportunities to be a part of emerging genetic medicine. And this has come through serving the OTS Society and its members, helping to build a gen med platform at Lilly, and now branching into an individual enterprise since June of last year. And so I'm so grateful for the years at Lilly and the opportunities I had to grow scientifically and to work alongside a lot of world-class scientists. Through many seasons of change, I learned so much about what makes a great leader. So the icing on the cake this last year was the opportunity to teach cell and molecular biology at Taylor University. And I've found that teaching and mentoring the next generation is a life-giving endeavor. So I thought I would talk a little more detail about my journey into genetic medicine. And I started at Lilly in 1994. And when I came, it was mostly a small molecule company. And I began working on raloxifene, what we call the selective estrogen receptor modulator, to prevent osteoporosis. And I was very excited a few years later, after joining Lilly, to learn about emerging ASOs and the early partnership that Lilly had with Ionis. Lilly took several ASOs to the clinic for cancer, but given that it was early chemistry and a challenging indication, the desired outcomes weren't achieved, and Lilly exited that space for quite some time. But my first oligo meeting was in 1997 in San Diego to learn about the promise of the technology. Then I spent several years working on the development of a peptide fragment of parathyroid hormone for osteoporosis treatment. And finally, I worked in our biologics department, developing therapeutic antibodies for diseases like cancer, immunology, cardiovascular disease, kidney disease. And the transitions between all of these modalities and disease indications was not an unencumbered path, really. It looked more like this. I'm showing here the Gartner hype cycle that describes how innovation emerges with enthusiasm and expectation and ebbs with challenges and uncertainty. And so, after decades in a large pharma company, these progressive stages actually look a lot more like this, can sometimes feel like a roller coaster, but it comes with opportunity to learn resilience in times of change. So there were two key events influencing the trajectory of my path to genetic medicine. It was the discovery of RNA interference and the completion of the human genome. So I spent several years then in a functional genomics group trying to explore this new space, looking at new targets and validating them in disease mechanisms for the portfolio. So when small molecules and antibodies were the major source of the therapeutics that we were working with, the druggable genome was quite confined. And RNAi screening was used to help direct us to genes that we could antagonize with small molecules or antibodies. So you can imagine the excitement to think about drugging the entire genome with emerging modalities. And I love this figure here that shows the progress and acceleration of disease gene identification during this era. And I think it's something to be proud of as a scientific community. In a couple decades, more than 4,000 genes have been identified for rare diseases, and up to 80,000 variants have been associated with complex diseases. And so I think one benefit of all this work that's really leading to speeding innovation in the gen med space has been the innovation in DNA sequencing, reducing the whole cost for looking at an entire genome. And it's amazing the dramatic improvement, outpacing even Moore's law. And as I checked just yesterday out of curiosity, the price is, I think, $3.99 starting for a whole genome sequencing, which is quite amazing. So while I led the RNA screening projects across the portfolio, we did try to deliver these unmodified sRNAs in vivo, and you can imagine we didn't have a lot of success. And as all things cycle, RNAi screening was displaced with other discovery tools like CRISPR. But that experience with RNAi left me curious and eager to follow the literature to see what could be possible to solve the challenges to make these RNA therapeutics appropriate as therapeutics. And these challenges could be summarized here in terms of increasing the potency, the durability, specificity, reducing off-target effects, reducing immunogenicity, and really enhancing delivery. So it was really quite a list of things to overcome. So it led to a period of reading and self-learning for many years on how these problems could be solved. And so these are some of the things that I read. I read a body of literature reflected in this excellent 2017 review by Anastasia Karova and Jonathan Watts. And I learned how the modification of the 2' position on the ribose could stabilize, increase the durability, reduce immunogenicity, as well as focusing on replacing the phosphodiester with a phosphothiowate bond, and then attaching targeting ligands that could take it very efficiently to tissues. So many of these modifications solved many of the problems for both siRNAs and ASOs. So other very meaningful bodies of work came from the entire scientific community at Ionis. This is a great review of what was done in the antisense space. Really appreciate all the reviews that Sudhir Agarwal wrote in antisense space. And I also discovered the Nucleic Acid Therapeutics Journal, which is the official journal that's sponsored by the Oligonucleotide Society. For siRNAs, the body of excellent work published by Alnylam that showcases the progression of their siRNA design addressed all those chemistry issues that I just mentioned. And their work was incredibly impactful to me. So one thing that I discovered in all of this reading is that these authors were part of the OTS Society and were presenting at an annual conference every year. And so I was able to start attending in 2018. That was my first meeting, and I was so impressed with the community, the data-driven focus, the willingness to connect and to share. I got hooked, and I haven't missed a meeting since. And so I'm going to take the opportunity to slip in a shameless plug for this year's meeting in Budapest, and registration is open. All right, so this is also a figure borrowed from a review paper from some Alnylam scientists that highlight the key advances in the development of RNAi therapeutics over a 20-year period, and how these increased from more rare disease indications to larger patient populations. And it's led to several FDA approvals. So if you're brand new to oligonucleotides and you looked back at this linear timeline, you might get the impression that this was a straightforward journey, and perhaps that it was an easy thing to do, but that wasn't the case at all. There were significant setbacks and challenges in developing these innovative solutions to enable oligonucleotide therapeutics. And so with the highs and lows then of enthusiasm and investment too that came over that 20-year period. And so in that 2017 review that I just mentioned from Anastasia Karova and Jonathan Watts, they brilliantly mapped this 20-year plus journey for both ASOs and siRNAs onto the Gartner hype cycle. And so many of us have used these and returned to these diagrams, but it really reflects all of the changes, the hope and the promise, some of the difficulties. So I want to point out that, around 2002, there were a couple of ASO failures in the clinic after a lot of enthusiasm about the promise of the modality, and the enthusiasm shifted to RNAi that was just discovered a few years before. However, RNAi would catch up with their own failures and challenges in clinical trials. But 2002 then was an important year. This was the time that the idea for the Oligonucleotide Society came to be. And as Art Krieg so eloquently highlighted in this video commemorating the 20th anniversary of the OTS, this is on the website. You can listen to it from the people that were there at the beginning. The society was conceived during this backdrop of clinical failures and real uncertainty if there would be a successful translation of oligonucleotides to therapeutics. So, having weathered a few of these cycles myself, I know that it takes real scientific leadership to persevere during a period of setbacks. And so, the founding members' vision and commitment to work together to advance the field during a time like this was really inspiring to me. So after 2002, the society was initially run by members whose time was divided with professional commitments. And the decision was made in 2009 to bring in a group of individuals who would organize the annual meeting. So Alexis Seca and Jerry Beatty of Event Innovations over the ensuing years professionalized the society and transformed the OTS into the multifaceted society that we have today. And so their contributions have been invaluable to the growth and the impact that the society has today. Because the OTS was so impactful to me professionally, I applied to be on the board of directors and was able to join in 2022 to help out with the work of the society. And if you're new to OTS, you might not be familiar with everything that's going on behind the scenes. So I thought I would take the time to quickly highlight that for you. Alexis Seca is our executive director right now, and she really is at the heart of the society keeping things going and running smoothly. So the board of directors typically serve on one or more of the committees that support the following areas of the society. So we have our annual meeting every year. There's a tremendous amount of work that goes into that all year long from a pretty large committee to bring that forward for everyone. Recently, we've added meetings throughout the year that we support locally and regionally. And there's a committee that reviews these applications and funds them. There was a wonderful meeting that happened in India this year, and there have been a few around the US, and we're really excited to continue to expand that. If you haven't been to the website, you should visit it and explore it because there's a wealth of new information and articles about what's happening in the space, and a lot of information is archived there. So the society supports the official journal, the Nucleic Acid Therapeutics. We have board members that are editors, and the journal makes available to the members periodically free articles, and you can access this on the website. So an exciting part of the society is to recognize the achievements of its members and encourage students and young career professionals in their journey. So if you're a student, check out our travel grant program for assistance to the annual meeting. We're really excited to be able to help students get to OTS. Another exciting program that launched the last few years is our mentoring program that matches board members and society members with early career scientists to share experiences. And the learning goes both ways. I've been part of this, and it's just been wonderful. I learned so much from the people that are the mentees that I interact with, and then they're friends for forever. We have education and outreach committees, and this is an area that I would love to see grow in the coming years. I think there's a lot of fear out there with genetic medicines in the general population due to a lack of understanding and about how they work, false information. And so as scientists and members of the society, I think we should actively and graciously educate others to help them understand the opportunities and the challenges of these new medicines. And lastly, but very importantly, there is a job board to connect companies with talent in the society. So if you're a company or somebody looking for an opportunity, please check that out. All right, so here is the board of directors in the Montreal meeting in 2024. Such an honor to serve alongside these world-class scientists to advance the mission of the society. I wanted to recognize the dedication of David Corey and Richard Geary sitting there in the front for their passion and commitment to lead the society and carry on the traditions that we have for excellent science and collaboration between industry and academia. All right. I'd like to transition now to making some observations about what's happening in the oligonucleotide field and where the interest and the discussion is focused. I was able to attend TIDES a couple weeks ago, and I think most of us will agree that there's really a lot going on in this space. And so much so, sometimes it's helpful to use databases such as Beacon here from Hansen and Wade to track changes and progress. And so I'd like to thank Ioana and Maddie for sharing some of these updates in their most recent newsletter. There's a remarkable number of oligos in development from the earliest discovery phases into clinical. So the Beacon database is tracking more than 2,000 oligonucleotides. And these break down into over 1,000 molecules in the RNA interference category, which can be siRNAs, microRNAs, some unspecified or small hairpin RNAs. Nearly 600 oligonucleotides, some again unspecified, over 100 aptamers, and 34 small activating RNAs. So it's pretty remarkable that the activity has grown so much in recent years. If you look at the breakdown of these oligos based on disease categories, neurological disorders rank number 1, followed by cancer and genetic disorders. And if you were to combine metabolic and cardiovascular siRNAs, they're a strong third at 262. If you look at the more advanced molecules that are progressing into trials over the last few years, you can see that there was a huge increase in 2024 over 2023, and the numbers for 2025 are looking strong as well. So by disease area, genetic disorders had 51 trials initiated last year. Neurodegeneration had 2. And again, if you combine the metabolic and cardiovascular space, it would be first at 57. So these increasing numbers in the cardiometabolic space, I think, reflect the robust delivery of GalNAc conjugated oligonucleotides and how well they deliver to the liver. And then the fact that the liver is this central organ regulating lipid metabolism and affecting cardiovascular health. So for managing cholesterol through LDL lowering, statins remain the standard of care. I had numbers from 2022 that there were 27 million patients in the US that had a statin prescription. So the goal to help reduce cholesterol is to reduce LDL to less than 55. That's really desirable. But for some of these patients that have very high, even achieving a less than 70 mg per deciliter is a goal of clinical trials. So even when patients are on statins, they reduce their cholesterol, they're still at risk for something that we call MACE, which is a major adverse cardiovascular event. And these can be uh, include a death related to cardiovascular, myocardial infarction, stroke, unstable angina requiring hospitalization, or coronary revascularization. So these are typically the adverse events that are tracked with patients that are on some kind of therapy for lipid lowering. So for some patients, being on statins or even the max statin treatment, the next line of therapy is inhibiting PCSK9. This is an enzyme that cleaves LDL and really impairs the ability of the body to internalize and metabolize cholesterol. So there are a couple antibodies that are approved for that, and one of the landmark siRNAs for a major indication was an siRNA from Alnylam, Leqvio. And so these then are meant to help reduce LDL further and help prevent these adverse events. But there's still residual risk with treating or just pursuing LDL lowering, and triglycerides have been identified as a risk factor. And some beautiful genetic targets have been identified in recent years, including angiopoietin-like 3, ApoC3, and Lp(a). And they're all expressed in the liver, and they're perfect targets for oligonucleotides to inhibit their expression. Then finally, this last piece of cardiovascular health, controlling blood pressure is so important, especially if you have diseased arteries and you're stressing them with uncontrolled blood pressure. There are approaches to reduce angiotensinogen, which is expressed in the liver and is this major activator of blood pressure. So this is the landscape, an amazing repertoire of genetic medicines to try to improve cardiovascular outcomes. All right, so the Beacon database is also very helpful to see all the activity around these targets. And I made these slides so they look less professional than if Maddie or Ioana had made them. So forgive me, but whoops, go back here. You can see that for angiopoietin-like 3, this was one of the, I think, the earlier strategies. There is a phase 3 molecule from Arrowhead. Ionis had an ASO that they took all the way to clinical trials, but stopped development at phase 2, seeing at the higher doses some undesirable impact on the liver. But there are still molecules out there in phase 2, phase 3 from Arrowhead and Lilly. So it'd be interesting to watch what happens with this target. So ApoC3 has seen a lot of exciting progress. In December of 2024, olezarsen from Ionis was approved to inhibit ApoC3 for hypertriglyceridemia. Richard Geary talked about this a few weeks ago in his webinar. And so there are other molecules in development as well that it will be interesting to see these come forward. Finally, Lp(a) has seen a lot of readouts and clinical trials, which is very exciting. Beacon highlighted this in their April newsletter and allowed us to share it here. Each of these molecules are unique in terms of the dosing range, the magnitude of reduction of LP little a, and um, uh, the number of patients that have been tested so far. So I think we're really eager to see how these molecules perform in these later stage clinical trials. All right, so this last piece for blood pressure is a very active space right now. And Alnylam is, I think, out in the lead in terms of developing an siRNA, but Ionis has an ASO that's coming, and there's a lot of activity in the preclinical, early phase 1. It'll be exciting to see where this goes. All right. So I thought I would come back to this first siRNA from Alnylam to treat PCSK9 in a large patient population. Often when you're leading, you have to face the headwinds yourself, and there's no opportunity to take advantage of drafting. Like, if you're racing and you're behind the leader, they take the headwind and you can benefit from that. So Leqvio was approved in 2021, and I've been very excited to see as this launch, how is the medical community fitting in this new modality for cholesterol management. So you can see from this investor slide that there is a significant lowering of LDL sustained with minimal doses for a very long time. They did look at outcomes with the percentage of these adverse events over time, and there looks like there is a very significant 2% reduction from placebo alone. This certainly would translate to saving patients' lives. But you can see that as this launched, and it's a new modality. The cost is, I think, around $3,500 a dose. I'm not an expert here, but getting 2 or 3 injections a year would be quite a bit compared to a generic statin. And so often payers, health insurance coverage, they want to see that really translates to improved patient outcomes. They're going to invest this additional money. Does it really keep people out of the hospital from these events? And is it safe? And so evidence then of this transition time to adopt this into healthcare practice, I think, is shown if you look at the revenue from Novartis that's marketing this drug. And Leqvio isn't in the top 10 even in 2023, but it's exciting to see that while I looked to see what the growth was in 2024, they did report 114% sales growth, and analysts are predicting that this could be adopted more rapidly in the next few years, and that we could see the prescriptions for this going up. I think the exciting data is that this has been in patients for 6 years or longer now, and there are reports of, just excellent safety, nothing in the kidneys or liver, and so it's exciting to see. And I think hopefully that this will set the stage for this other wave of products coming for triglyceride lowering, and that the adoption and the translation into clinical practice and getting these to patients will be easier. So Again, here I'm borrowing from Alnylam's story as they're leading and out front with an siRNA for angiotensinogen to reduce hypertension. You can see with knockdown, they have significant blood pressure lowering sustained for a very long period of time. But what I love that they're taking the time to do with their partner is to do these large patient clinical trials. That the outcome and the measure in phase 3 are a measurement of these MACE type endpoints, and they hope to launch with evidence that these medicines really have outcomes that improve patients' lives, reduce costs. I think the only challenge, right, to invest in these large outcome studies is that you need large numbers of patients, and they take a long time to see these adverse events either accumulate or improve. So it'll be some time before we get the data from these outcome trials. All right, kind of abrupt shift here. I thought I would shift gears by throwing out some statements or some questions that we could take into the question and discussion period. So I think one question that people ask a lot, new to the field, or even over time, isn't siRNA better than an ASO versus RNA editing, et cetera? And I think that the answer is that most companies will need to be fluent in many modalities if they're committed to a disease area. And the right target comes along that's really critical to change the trajectory of the disease. It may be limiting if you're committed to only one modality. And I think you see this a lot. Big pharma are adding in all these different modalities. Smaller companies, too, that maybe are specializing in one are trying to get expertise. And what follows on is that once you do that and you have all these modalities at your fingertips, then you can combine them with different molecules that may enable delivery to new tissues. And examples of this that are emerging, some of the best ones are these antibody peptide small molecule conjugation to siRNAs and ASOs. All right. So I want to just come back to a benefit of this era of building these databases of genetic diseases, and how this reduced cost of sequencing is enabling us to dial in genetic medicines for an individual. I think some of the first work that inspired me to rethink this way was BioNTech taking patient tumors, sequencing the RNA, and finding unique mutations that were driving cancers for these people, and then making mRNA vaccines to present these neoantigens, and you would get a personalized vaccine for your cancer. I remember just thinking, wow, this is amazing. So one of the first people to envision that for oligonucleotides was Dr. Tim Yu, who was made aware of Mila, who had a rare mutation that was unique to her. And he thought, why couldn't we make an ASO for her disease? And not only did he think about doing that, but doing it in a very short time frame. And within a year, people came together and rallied around this concept and made a medicine for her. And I think it taught all of us how we can re-envision this timeline for discovery and what we can do. There is a group, the N of One Collaborative, where many people are trying to expand this concept. And there's a great website if you want to get involved, or sponsorship, because it's not for profit. Please check out the website. And I'd like to recognize Stan Crook, who's been involved in the development of ASOs from the very beginning. And I love that at this point in his career, he's dedicating his time and his talents to help these rare disease patients too. And so he is leading the N. Lorem Foundation, not for profit. They have a meeting every year that's very informational. You can go to their website, think about sponsoring this work as well. And then I think most of us have seen in the recent news this idea coming forward for gene editing and personalizing and editing molecule for baby KJ. And it was done in 6 months, which is astounding. And so Fyodor Ernov has been involved in this. I follow him on LinkedIn. I love his comments, and I'm quoting him here from LinkedIn. He said, "Let's get this done, people. E pluribus unum, out of many one." So I'm really excited to see what comes in the near future in this space. So what have we learned from these individualized medicines? I think that we're faster when we collaborate. I think we can move fast when we're committed and passionate and try to remove barriers. Thinking this way, I think, will move us all to think about shorter development times, help reduce the cost of these genetic medicines. Often these rare disease patients as well don't have time on their side, and we need to move very quickly for them. And I think we learned this too during the pandemic, when it was necessary to make vaccines and antibodies. That was done in less than a year as well. All right, so I think something that's going to be really critical to fully enable this modality for large patient populations. And even we can think about the rare disease community as a large patient population, even though they may be an individual or a small group of people. I think the estimates are that 10 million people worldwide have a rare disease. And so it's estimated that a possible 10 tons of material might be needed within 10 years to meet the demand for therapeutic areas like hypertension, obesity, MASH, Alzheimer's, these cholesterol and triglyceride diseases that individuals have. There's millions of people that can benefit from it. So the major way to make oligonucleotides now is through chemical synthesis, and the community knows that it takes an unsustainable consumption of solvents to make this happen. And so there's some exciting work that was presented at TIDES by Hamjin and Bakem, showing that by using enzymatic ligation of smaller fragments that you can get a higher yield from, that you can get longer products at high purity. And albeit the smaller fragments are made by chemical synthesis, but I think there can be some savings and efficiencies with this methodology. I think what's really required to transform this space is what Codexis is working on, to engineer an enzyme that can extend and polymerize a new oligonucleotide on resin. And I think that we need to recapitulate what we saw in that genomics era, where the the cost per ton of oligonucleotide really needs to come down. If this happens, then we can afford to help more rare disease patients and take this forward into large patient populations. So one thing that I think about, and I would love to hear more from the community, because I really haven't seen much research in this area, but maybe it's out there. But we do know that when large patient populations consume pharmaceuticals and you excrete them, that they can end up in the environment and in aquatic ecosystems. And here's just a list I got from a paper of some of these massively consumed products. And there are recommendations now not to, how to dispose of your pharmaceuticals. So these oligonucleotides that we're making for year-long durability have really good stability. And so I think there's limited evidence that these are being excreted in any substantial quantity to get into the environment. But I think that I would love to see us as a community be mindful of this, and if these really scale to these massive quantities, understand if there's any environmental impact of using synthetic oligonucleotides. All right, last point here as we wrap up. I've talked about these living through, cycles of excitement and investment and, contraction of that investment. And I also snatched this off of LinkedIn from Biospace that shows nicely the recent cycle of investments coming into biotech. And probably many of you have experienced this downward trend to significant reduction in investment. So I know that these cycles, especially one like this, is multifactorial. You probably can't point to any single thing, but it made me reflect. And is there, based on what happened with the Oligonucleotide Society during their cycle of expectation and investment and contraction, is there an inverse relationship between the influx of investment dollars and a reduction in innovation that it just inevitably happens due to the IP that needs to be protected and is siloed during this project or process of investment into companies. So, could the trajectory be changed if this is true? As money goes in, is the learning and the innovation inadvertently slowing down? Does it actually cause maybe a bit of this contraction, before it recovers? So I just wonder, in the case of the OTS, where they came together and collaborated and learned over non-competitive information, could that change the rate of or kind of the magnitude of the contraction and then the slope and the rate of recovery? So just something to think about and to encourage us as we continue to push these genetic medicines and reach or seeing new opportunities, but also new challenges, that we remember how the oligonucleotide field came out of a period of challenge. And so I think that it's an example to us all for the history books and worth retelling and remembering during the next cycle of innovation and the challenges that are ahead. So I extend an invitation to you to come and be a part of the OTS community and this mission to between academia and industry to collaborate, to innovate, to communicate, to educate others. Um, Open it up for questions and comments. I think we still have some time. I did go a little bit longer than I planned. But if we don't have time to answer your question, feel free to reach out to me on LinkedIn, and I'd love to meet you.
Yes, thank you, Rebecca, for this excellent overview of all the research that has been done, that has been going on, but also about the challenges for the near future, but also for the slightly longer future. So maybe to start with a question on that. So, do you think that we have the right infrastructure? Because if I see how many patients are eligible for these kind of treatments, what is needed if you think about infrastructure and the need for that?
I do think that probably the most critical area for innovation is in synthesis to really drive the cost down because we as a society own the cost of these medicines, right? We all contribute to the dollars that are available either through the government or privately held healthcare insurance. So we do have to manage that and think about it. So finding sustainable ways that won't impact the environment to produce oligonucleotides. Um, I think is the key point. And I know there's a tremendous, I'm not a manufacturing person, but there's a tremendous amount of effort, right, to build the capacity to prepare for these larger patient populations. We're not there yet. I think that will be a bottleneck until it's figured out and we're at this next level of the ability to produce oligonucleotides very cheaply.
I think that maybe to add one thing for now. So if you have a question for Rebecca, please use the Q&A. So and okay, well, let's say okay, we now solved the whole synthesis problem, so we can synthesize as many compounds if we want. So what about the treatment? Do you think that the infrastructure in the hospitals, maybe at the GPs or at
Mhm.
the local institutions, is enough that we can also treat all these people, or do you think that we also need some education in that?
Yes, so education, I think, is huge. As I mentioned, I do think there's a lot of fear about anything that we say is genetic or gene editing and oligonucleotide. We saw that with the vaccine, right? When we talked about nanoparticles, that sounded like science fiction, I think. So education to um, uh, and we still have ways to go to make these completely safe, right? So we do need to communicate the opportunity and the challenges. Um, our delivery needs to become more sophisticated. So in the case of targeting the liver, this is I think very easy and it's a sub-Q injection. People can probably administer it to themselves with an auto-injector. But if it's IV and you have to go to an infusion clinic for that, that's going to be very expensive and costly. So even that, I don't think is sustainable long term. We need to have a breakthrough in CNS delivery, which we might be on the verge of with some really promising data for antibody oligonucleotide conjugations. And if you can deliver through sub-Q injection peripherally and get molecules to, say, the nervous system or other places, then I think that will be easier. But to do IT injections or direct injections into the brain, that is not sustainable too. I think that repeated IT injections will be very difficult for a lifetime for a person. So yeah, that's an area for the whole field to grab hold of and solve. It will come through delivery. Mhm.
Yeah, I think I agree in that. So I think for the liver, of course, we have the gallneck that works pretty okay. For the CNS, we have the IT injections. In the eye, we can do injections that stays local. But I think also the delivery part is quite a big challenge in the
Yes.
field.
It needs to all move sub-Q. Mhm.
Yeah,
Or oral.
that
That would
that would be
be
perfect. Yes.
that would be perfect.
Some questions from the audience. So a question from Michael. Um, of molecules in the oligo discovery pipeline, what are the characteristics of those that progress to trials? And he's working in the preclinical field. So can you give any specific characteristics that kind of compounds that are more likely to move into a clinical stage?
Yeah, so I go back to that slide on kind of what the challenges are. You need to have the most potent molecule possible so that your dose can be small, reduce off-target effects. It needs to be stable and survive the journey from the injection site to the target of interest. So the chemistry has to be um, optimized without jeopardizing potency. And then in combination with all of that, it really needs to be on target. And so I think the chemistry is so important to stabilize the molecule and um, get it to the tissue. There's a lot of biology that also RNA biology that goes into the right siRNA. It needs to target the right region of the gene and have accessibility. And I think that can make a difference as well.
Yeah, and a little bit of follow-up question on the delivery part. So a question from Gohan. Is the low efficiency of oligo delivery into the cell still the leading cause of clinical failure?
Um, I think that certainly contributes to it. So if you are not successfully engaging your target and knocking it down, that can lead to failure. But sometimes there are incorrect ideas and hypotheses that inhibiting that target is actually, and you may knock it down, but you may not see an improvement in the disease. And it's not necessarily that you're knocking the gene down, but that inhibition in the disease context actually moves the needle for the patient in a way that changes the outcome. And so, it's a combination. You want the best molecule, but even when you have that, then you need to make sure that in the disease context, that it's going to be beneficial to the patient. And sometimes we're wrong, right? Biology always has surprises.
Yeah, I think this is why we always have to test the hypothesis. So we still have a few questions open. So here it's on the hour again. So do you have can we have a few short answers
Sure.
to the questions? So they might be slightly longer, but so the first one is really challenging to give a short answer, I would say. But let's see how far we get. So can you share your perspective on splice switching oligonucleotide as therapeutic? And its future and the future of the splice modulating oligos.
Sure. I mean, it's so exciting that they actually work. And in the rare disease space, I think this is where they're of highest value, right? But it's not like it's a catalytic activity like AGO2 is, where you have a very small amount that can target the same gene over and over again. So, and then often too, they have to get to the nucleus. So, you may need a higher amount for it to work. But sometimes in the rare disease, whatever the mutation is, it's just not amenable to splice switching, right? So I think, that's an unfortunate limitation too, that, you really have to understand the disease and if you can even use that technology. But I think potency and durability are a real challenge. And I think most of them now are being delivered by direct delivery into the CNS. And for people that have no other option or hope, then absolutely it's worth it. But ideally, transitioning to IV with some kind of delivery to get the splice switching oligo in the cell into the nucleus would be great. But I'm not sure, if you can get enough in there. So hopefully this will be the next wave of innovation in that space.
Yeah.
With direct delivery.
So to continue on that, so you mentioned already the delivery into the brain. And again, a very short answer. So what would be are your thoughts about the mechanisms of delivery over the blood-brain barrier? What, in your opinion, would be the best strategy for that?
Well, I was listening to a lot of talks at Tides and statements that people made. There's transferrin is uh, likely the next GalNAc. It works, right? It's expressed on the endothelium. It can take things across the blood-brain barrier and into the brain. And so that's very exciting to see, so many companies using, different ways to target the transferrin receptor. So are there others? Maybe. But I think that the transferrin examples that are out there in the industry are very exciting.
I agree on that. So a question also on the beacon data that you showed. There are way more siRNAs into the preclinical and clinical development than ASOs. So any thoughts on this?
Yeah, so it kind of goes back to being fluent in all modalities. I think what's attractive about RNA interference is maybe the durability, but ASOs are coming along. I think a lot has been done to mitigate off-target effects of siRNA, but given their ability to have microRNA-like targeting, you have to spend a lot of time characterizing the off-target gene signature and mitigating that if possible. So, so I think the durability piece is what's most attractive about siRNA. But the ASOs have their place too. Maybe you don't need that amazing durability. Maybe that durability is a liability in certain cases. And if you listen to Richard, his webinar, our current president and from Ionis too, he said something very similar, right? That it really depends on the target, the tissue that you're in, the thing that you're trying to do. ASOs can do things that siRNAs can't. So I love them both.
Very last question for the webinar. So can you comment on the high dose? So especially compared to small molecules that's needed for oligonucleotide therapeutics. And might that also be a reason for the failure of some of the clinical trials?
Well, early on, that was definitely the case. I would say that without the stabilization, there were pretty high levels. But actually, with GalNAc conjugates, there's just like a microgram or, very small dose that's once or twice a year, it's taken up and stored in the tissue. So when you dial in the chemistry and the delivery, you can really dial back on the dose. Maybe for the splice switching oligos and things like that, the dose gets high. As the dose goes up, yes, the off-target effects will increase, having some toxicity, maybe immunogenicity, all of those liabilities increase. So having the best molecule is really important to make sure that it's efficacious and safe.
Good. Thanks a lot. So then I give the word back to Erin.
Great. Well, thank you, Rebecca, for that it was really insightful, the webinar, and it was good to see your career trajectory. It was really good to have some questions answered. I know that there were still a few questions outstanding, so you can reach out to Rebecca via LinkedIn. She's easy to find on there. And I just would like to thank you once more for giving such a great talk and remind everyone to keep an eye on the OTS webinar website for more webinars that are coming up.